के बाद।
हाँ, जैसा भी हो। तो मैं तेरे को पहले फाइनली मतलब फॉर्मली इंट्रोड्यूस कर देता हूँ। So for today's talk, we have Dr. Buddhi Prakash Jain. He is working as an assistant professor at Department of Animal Sciences, Central University, Bihar. He has pursued his PhD from School of Life Sciences, Jawaharlal Nehru University, and secured gold medal in his master's program in University of Rajasthan. He have published 4 books and have many national and international publications to his credit. His thirst area are cancer biology, endoplasmic reticulum, and stress biology. So now I invite Dr. Buddhi Prakash Jain to kindly deliver his talk.
Thank you, Dr. Arun. Once again, thank you, Dr. Arun, for giving such a nice introduction. So today we are going to talk on a topic that is SG2NA, a scaffold protein with role in the endoplasmic reticulum. Stress. Briefly, we will discuss about this protein, what this protein, and how this protein involved in the endoplasmic reticulum. Stress. So first, as an introduction, we will understand about this protein. This protein SG2NA, this protein was initially identified as a nuclear autoantigen in lung and bladder cancer patient, and its expression increases during S and G2 phase of cell cycle. That's why it was named as SG2NA. This S and G2 denotes the cell cycle phases, and Na is the nuclear autoantigen. So this protein shows a cell cycle regulated expression. This protein belongs to a striatin family, and the other members of this family are the striatin and the zynidine. And this striatin family comes under the WD40 repeat superfamily. And because of the presence of this WD40 domain at the C terminal, these proteins act as a scaffold protein. And what is the meaning of scaffold protein? The scaffold proteins are those proteins which act as a which provide a platform for the assembly of the multiple proteins into a single complex and then mediate the downstream effect. So these scaffold protein basically act as a platform for assembly of the multiple proteins. So besides the presence of this WD40 proteins, the other domains are also present in the striatin family, like at the N-terminal, Cavalion binding domain, then the coiled domain, and the calcium dependent calmodulin binding domain. So this is the domain architecture of these striatin family proteins. Coming to the functional part or the other literature of this protein, this striatin and the SG2NA, they form a regulatory subunit of the protein phosphatase 2A. Protein phosphatase 2A basically have 3 subunits. One is a catalytic subunit, one is a regulatory subunit, and one is the structural subunit. So this striatin family act as one of the type of regulatory subunit for this protein phosphatase 2A. The other function of this SG2NA, that like N-terminal region of the SG2NA act as a transcriptional activator, and while the C-terminal of this protein had an inhibitory effect. So in our earlier studies, we have established that this SG2NA protein undergo alternative splicing at the exon 7 to 9 and generate the multiple splice variants which have the tissue restricted expression and the growth responsive properties. And also this among these among the 3 members of this striatin family, the SG2NA was first to arise during evolution. This SG2NA protein also interact with the DJ1 and the AKT protein in the mitochondria and the membrane and protect the cells from the oxidative damage. And regarding to the cancer cells, it is established that this under a protein DJ1 and the activity and activate the AKT. So this is the literature till now, the literature available regarding this protein. We will also understand the isoforms of this SG2NA that I told you earlier, that SG2NA generate multiple splice variant by the alternative splicing at the exon 7 to 9. So these are the 5 isoforms which are generated by the alternative splicing as well as RNA editing and intron retention. So 87 kilodalton is the isoform of the SG2NA which have all the 18 exons, means there is no splicing, alternative splicing, all the 18 exons are present. But in case of 82 kilodalton isoform, exon 9 is spliced out. So exon 9 is absent, and one more isoform, that is 78 kilodalton, which is generated by the splicing of the exon 8 and 9. So both exon 8 and 9 are absent. Then one more isoform, that 52 kilodalton, which is generated by the RNA editing in the transcript of the 82 kilodalton transcript. So this leads to the premature termination of the translation and generation of a smaller molecular weight isoform, that is 52 kilodalton. And 35 kilodalton is also generated by the intron retention after the exon 7, and because of this intron retention, again this leads to the premature termination and a generation of a smaller variants of the so these are the isoform architecture of this protein. Coming to the present work, so what we wanted to do, we wanted to know further the functional aspect of this protein SG2NA. So for that, what we did, we first downregulated this SG2NA protein by using the shRNA in the NIH3T3 cells, and we confirmed the downregulation of this SG2NA by the Western blotting. So first we downregulated, or we knocked down this SG2NA protein in the cell in NIH3T3 cells. And after that, what we did the total proteome analysis, means we analyzed the total proteins, what are the proteins that get modulated upon SG2NA depletion or the SG2NA knockdown. For that, we did the 2-dimensional gel electrophoresis followed by the Mardi-Graph analysis. So in these experiments, what we found that upon depletion of SG2NA, 110 proteins get modulated, 55 get upregulated, and 55 get downregulated upon depletion of SG2NA. And out of 110, we were able to identify 30 proteins, that 20 proteins that get upregulated, and 10 proteins that get downregulated upon SG2NA knockdown. So these are there is change in the proteome upon depletion of the SG2NA. Upon further analysis, what we found that out of 30 proteins, a group of proteins, so these are the group of proteins out of 30 proteins which are modulated upon SG2NA depletion. They are related to the protein folding and the endoplasmic reticulum stress inside the cells, like protein disulfide, phospholipase C, GRP78, HSP90. These are the proteins which are like elongation factor 2, polyglutamine binding protein 1. They get downregulated, and these proteins get upregulated upon depletion of SG2NA. So we hypothesize that might be there is a disturbance in the ER functioning, and there is an induction of ER stress upon depletion or knockdown of the SG2NA. Whenever there is an if we are depleting the SG2NA, there is a disturbance in the ER functioning, and there is a subsequent induction of the ER stress. So up to this point, we hypothesize that this SG2NA might have some role in the ER functioning. Before going further, we will also understand what is ER stress and what are the consequences of the ER stress. So this is the slide. We can understand that what is the ER stress and what are the downstream effect of the ER stress. First, what is ER stress? See, endoplasmic reticulum. Endoplasmic reticulum is the cellular organelle where the proteins are folded properly and various post-translational modifications occurs, and after that the proteins are targeted to the different locations inside the or outside the cells. But whenever there is a disturbance in the protein folding process, maybe there are the several factors like the calcium imbalance, maybe glucose deprivation, maybe some viral infections, maybe some disturbance in the redox homeostasis. There are the several factors which disturb the protein folding process. And when there is a disturbance in the protein folding process, the misfolded or the unfolded proteins, they start accumulation inside the ER lumen. Means the unfolded or the misfolded proteins, they accumulate in the ER lumen, and this condition is known as ER stress. The endoplasmic reticulum is in this stress condition. Now, what are the consequences inside the cells to restore the homeostasis inside the cells? These unfolded or the misfolded protein, they activate or initiate the downstream signaling, and that signaling is known as unfolded protein response signaling. The unfolded or the misfolded proteins, they initiate or activate the downstream signaling, and that signaling is known as unfolded protein response signaling. And there are the three branch of the UPR. There are the three branch or three activators of this UPR signaling. One is the ATF6. ATF6 is a transcription, is a transmembrane protein, is a receptors which is bound to the ZRP78. ZRP78 is a chaperones which assist in the protein folding. These receptors like ATF6, PARK, IRE1, they are present in the inactive form because GRP78 is attached to these receptors and making them inactive. But when unfolded or the misfolded proteins start accumulation in the ER lumen, they competitively bind to these receptors, and GRP78 now detach from these receptors, and these receptors become activated. And what these receptors do, they initiate, they mediate the downstream signaling, and that signaling is known as a unfolded protein response signaling. And what is the primary role of this UPR signaling? They try to restore the ER homeostasis. Means somehow they increase the expression of the genes which assist in the protein folding process, like chaperones, ZRP78. They increase the expression of ZRP78 so that the protein folding process can be further enhanced, and the load or the burden on the ER can be reduced. So the overall effect of this signaling is to restore, increase the expression of various UPR target genes which are involved in the protein folding, or they reduce the burden on the ER. So they try to restore the homeostasis. But the same signaling, if the ER stress condition is persisted and they are not able to overcome this stress condition, the same signaling leads towards the apoptosis. The same signaling which are trying to restore the homeostasis, now the same signaling is leading cells towards the apoptosis. Now how these three signaling work? First is the ATF6. When these ATF6 receptors become activated, they are transported to the Golgi complex, where they are cleaved by the two protein S2P and S1P, and this cleaved ATF6 now act as a transcription factor, moves to the nucleus and increase the expression of various UPR target genes like protein disulfide isomerase, GRP78, and etc. Other transmembrane receptor is a PERK. When this receptors become activated, first they autophosphorylate their cytoplasmic domain, and once this cytoplasmic domain become activated, what they do, they phosphorylate this EIF2 alpha. EIF2 alpha is a eukaryotic initiation factor alpha, which is required during translation process. So when this EIF2 alpha is phosphorylated, this EIF2 alpha become inactivated. When this EIF2 alpha is inactivated, there is an overall reduction in the protein synthesis process. So if there is a less protein synthesis, there will be less protein to become fold. So there is a reduced burden on the endoplasmic reticulum to perform the function of protein folding. So this signaling overall reduce the protein synthesis and subsequently protein folding. But this phosphorylated EIF2 alpha selectively increase the transcription of ATF4. This EIF2 alpha, although it's reducing to alpha selectively increase the expression of ATF4. Now ATF4 act as a transcription factor. It migrate to the nucleus and it increase the expression of various chaperones, antioxidant response shock, and other proteins which assist in the protein folding. And this CHOP protein, which leads the cells towards the apoptosis, this CHOP increase the expression of another protein, that GADD34. This GADD34 dephosphorylate the EIF2 alpha protein. So this PERK signaling basically try to restore the homeostasis, but if the stress persisted and they are not able to overcome this stress condition, the same signaling also increase the expression of various genes which are involved in the apoptosis process. Third signaling is the IRE1 alpha. When this IRE1 alpha receptors become activated, what they do, this activated receptor act as a endonuclease, and they cleaved this XBP1 mRNA. Means they remove a small fragment from a XBP1 mRNA, and this spliced XBP1 now act as a transcription factor for the expression of various chaperones, ER associated degradation pathway proteins, and various proteins which are helpful for the restoring the endoplasmic reticulum homeostasis or the ER functioning. So overall, these three signaling basically increase the expression of various target genes which are involved in restoring the ER functions or the homeostasis. So this is all about the ER stress and the UPR signaling. Coming to our experiments, from previous slides, I told you that we hypothesized that this SG2NA protein might have some role in the protein folding or the ER stress, because upon depletion of SG2NA, there is a increased expression or there is a modulation of various ER stress related proteins. So our next target aim to first validate our proteomic data, means whatever the data we are getting in our proteomics, two-dimensional gel electrophoresis and MALDI-TOF, whatever these results we are getting, so we wanted to confirm those data so that we confirm by the Western blotting, and we found that whatever the results we are getting, these the right panels, these are the dots from the gel, two-dimensional gel electrophoresis, and this is the Western data which is showing that in SG2NA knockdown condition, there is an increased expression of ZRP78, there is an increased expression of HSP90B, and there is an increased expression of other proteins like vimentin and PKM2, which we got in our proteomic data. So up to this point, we validate that whatever we are getting in our proteomic data is correct. Further, we want to confirm that upon depletion or knockdown of Ag2Na, there is an induction of ER stress and activation of various UPR signaling. For that, what we did, we also did the transcript analysis ATF4 CHOP by the semi-quantitative PCR as well as real-time PCR, and we found that yes, upon depletion of Ag2Na, there is an increased expression of various UPR target genes, which I told you that whenever there is a induction of the ER stress and activation of the UPR signaling, various UPR target genes get activated or get upregulated. So that also we confirm by the semi-quantitative as well as real-time PCR, this GRP78, GRP94, PDIA3, ATF4 and the CHOP, they get upregulated upon Ag2Na knockdown. Further, yes, we up to this point again we are sure that yes, there is an induction of ER stress and activation of the UPR signaling. But next we wanted to confirm whether all the three branches of the UPRs, they get activated or a particular branch is activated upon Ag2Na knockdown. So what we did, we checked the splicing of the XBP1, and we found that in Ag2Na knockdown there is a splicing of XBP1 which confirms activation of branch of the UPR signaling. We also checked the ATF6 level, and we found that there is a increase, there is a decreased level of the full length ATF6 and increase in the cleaved ATF6. So this further confirmed that there is an activation of ATF6 branch also upon Ag2Na knockdown. And in this picture, in this experiment, we confirm that whether this Ag2Na is localized in endoplasmic reticulum or not. So for that we use the tracker dye for the endoplasmic reticulum, and this immunocytochemistry for the Ag2Na, and the this merged image is showing that this Ag2Na is also localized in the endoplasmic reticulum. So now up to this point again we are confirmed that yes, upon depletion or knockdown of the Ag2Na, induce the ER stress and activate the UPR signal. Another experiment, what we did, we induced the ER stress. We induced the ER stress by using the ER stressors like thapsigargin, tunicamycin. This thapsigargin and the tunicamycin, these chemicals, they induce the ER stress. How they induce this thapsigargin? This thapsigargin is a inhibitor of ATPase, calcium ATPase inside the cells. So this if the calcium ATPase is inhibited, the calcium cannot be pumped or transported to the endoplasmic reticulum. The cytosolic calcium level increases, and the calcium level in the ER lumen decreases. So this decrease in the calcium or this imbalance in the calcium level affect the protein folding process and ultimately induce the ER stress and the UPR signaling. Tunicamycin is also assisted in the post-translational modification, and this tunicamycin also induce the ER stress inside the cells. So when we are treating with the various concentration of the tunicamycin and the thapsigargin, and we are checking the expression of GRP78, which is confirming that upon treatment with this tunicamycin and the thapsigargin, there is an induction of ER stress. But what is the effect of this drugs of tunicamycin and thapsigargin on the Ag2Na expression? Then we found that upon treatment with this tunicamycin and the thapsigargin ER stressors, the expression of Ag2Na goes up. So in earlier experiments, we are saying that if we deplete Hg2Na, there is an induction of ER stress. In another experiment, if we induce ER stress, there is an increased expression of Hg2Na. So this both the results further confirm that Hg2Na is Hg2Na is playing some role in the ER functioning. That's why if we downregulate Hg2Na, that there is an induction of ER stress. If we induce ER stress, Hg2Na level goes up. So they by some pathways they are playing some role in the restoring the ER homeostasis. So this upregulation of the Hg2Na, upon upregulation of Hg2Na, increase expression of Hg2Na upon treatment with this ER stressors, we also confirm by the protein level also, and we also got the same result like upon treatment with this tunicamycin, the expression of the Hg2Na goes up. Even in the Hg2Na knockdown cells, interestingly, we found that when we treat with this tunicamycin, the level of this Hg2Na goes up, is in expression increases. Similarly, when we treat with this thapsigargin, and at the different time point, the expression of the Hg2Na goes up in the control cells and even in the knockdown cells. So even in the knockdown cells, when we treat with this thapsigargin, the tunicamycin, the expression of the Hg2Na increases. In further experiments, we wanted to confirm that this increase in the expression at what subcellular location, means expression is increasing, but at what subcellular location this expression is going up. So for that we did the subcellular fractionation. We treat the cells with the tunicamycin and the thapsigargin, and at the different time point at 6R and 24R, we did the subcellular fractionation, and we found that this increase is mainly in the microsomal fraction. This microsomal fraction contains the endoplasmic reticulum. So this increase in the Hg2Na expression on treatment of these ER stressors are mainly in the microsomal part. Further, we also did some in vivo experiment where we inject this thapsigargin in the mice, and after 24 hours after the treatment, we did the this analysis of this Hg2Na protein in brain and liver, and we found that in liver this 78 kilodalton isoform goes up. The expression of this 78 kilodalton expression goes up upon treatment with this thapsigargin. So this is further confirming that upon treatment with this thapsigargin and the tunicamycin or the ER stressors, the expression of Hg2Na increases. One thing that in the cell line we are talking about only one isoform, that is 78 kilodalton, because in case of cell line only the 78 kilodalton isoform is present in most of the cell line. But at the tissue, if we talk about a tissue, there is a separate work which show that there the expression of different isoforms in tissue specific manner. Like here we can say that in brain the expression of 87 kilodalton is there, but in case of liver the expression of 87 kilodalton is not there. So in case of tissue, different isoform expressed in tissue specific manner, but in case of cell line only 70 kilodalton isoform is expressed. So moving further, next we wanted to check that the effect of on the cell viability, effect of Hg2Na knockdown on the cell viability under ER stressors. Means in the control cells and the Hg2Na knockdown cells, both the cells, one cell line which are control and one cell line where we have depleted the Hg2Na, Both the cell lines we treat with the ER stressors like thapsigargin and the tunicamycin, and we wanted to check the effect on the cell viability. For that, we did the MTT assay, and we found very interesting result that compared to the control cells, the SG20 knockdown cells show very less viability under ER stressors like thapsigargin and the tunicamycin. That clearly we can say that after treatment of this tunicamycin after 48 hours, the cell viability is very less compared to the control cells. And these are the control, like where we treat the cells with the 1 millimolar of glucose and the 5 millimolar glucose. So this experiment further say that the effect of this SG2NA in the cell viability under ER stress condition. We also confirmed this result with another experiment where after treatment, this is the control TG, means control cells treated with the thapsigargin, control cells treated with the tunicamycin, KD knockdown cells treated with the thapsigargin, and knockdown cells treated with the tunicamycin. And we treat these cells and we stain with the hex stain, PI staining, and these are the merged picture. So the cells which are stained with the hex show that these cells are the apoptotic cells, and the cells which are stained with the PI, they are the dead cells with the normal nuclei, and the cells which are stained with both hex and PI, which indicate that these are the dead cells with the apoptotic nuclei. So in this, we can clearly see that in case of SG20 knockdown cells and treatment with the thapsigargin and the tunicamycin, the number of cells with the hex and the PI stent cells are more. It means that if we treat the cells, if we treat the control cells with the thapsigargin and the tunicamycin, and if we treat the SG20 depleted cells with the thapsigargin and the tunicamycin, there is a difference in the cell viability. So this SG20 is playing some role in the cell viability under ER stress condition. The last experiment, we just wanted to check whether there is a we wanted to check whether there is a changes in the cell cycle progression after SG20 knockdown if we treat the cells with the ER stressors. Means we wanted to check the effect of ER stress on cell cycle progression after SG20 knockdown. First, we checked the expression of the cyclin D1, which is a main cyclin, which is required for the progression from G1 to S phase. So the expression of this cyclin D is less in SG20 knockdown cells, but and this we did with the flow cytometry. These are the results of the flow cytometry experiment. These are the different time points: 0 hour, 6 hour, 24 hour, and these are the control cells treated with the thapsigargin 1 micromolar. These are the SG20 knockdown cells treated with the thapsigargin 1 micromolar. These are the control cells treated with the tunicamycin 1 microgram per ml, and these are the SG20 knockdown cells treated with the tunicamycin 1 microgram per ml. So I'm coming to the next slide, which is the summary of these results. This is the bar diagram showing that how many percentage of cells in different phases, like control cells at the 0 hour, 6 hour, and 24 hour. These cells are this orange color is the sub G1 phase. This sub G1 phase are correspond to the pro-apoptotic cells, and these are the blue is the G1 phase, these are the S phase, and these are the G2 phase. So if we summarize these results, so this is the summary. Like control cells at the 6 hour and 24 hours, and these many percentage of the cells are in the subG1 phase. These are the G1-S phase, and these are the S phase, and these are G2M phase. So we can see that in SG20 knockdown conditions, the cells in the sub-G1 phase are more compared to the control cells. So this indicates that upon treatment with the thapsigargin in the SG20 knockdown cells, more and more percent cells are in the sub-G1 phase, which are pro-apoptotic cells. Similar results were obtained for the tunicamycin also, like at the 24-hour of the treatment in the SG20 knockdown cells, the cells are more in the sub-G1 phase or the pro-apoptotic phase. And also one more observation there, compared to the control cells, SG20 knockdown cells upon thapsigargin treatment, the cells are less in G1 phase compared to the control cells. Like in case of control cells, tunicamycin treatment, the 71.9% cells are present in the G1S phase, while in SG20 knockdown cells, only 55.18% cells are present in the G1S phase. So these results show that the cells under SG20 depleted cells under ER stress show different kinetics for the cell cycle progression. And we also check the expression of the cyclin D1 and cyclin D3 in control and the knockdown cells, and in that tunicamycin and the thapsigargin treatment, the expression is less compared to the control cells. So from all the above experiments, we have concluded that SG20 is a regulator of endoplasmic reticulum homeostasis, and its depletion leads to the ER stress condition. This SG20 also have role in the cell survival under ER stress. And SG20 knockdown cells have a different kinetics of the cell cycle under the ER stress. So these are the conclusions from the above. And now, what are the future perspectives? First is the this second one is the first mechanism of the involvement of the SG20 ER stress. So we have established that this SG20 have some role in the ER functioning and the ER stress and the UPR induction of the UPR signaling. But what is the mechanism? Means what are the interacting proteins? How this SG20 is playing role in ER functioning or induction of the ER stress or the ER homeostasis. So this is our next objective. And also another objective is that a role of STRIPAC in ER functioning. This STRIPAC, briefly I want to explain. This STRIPAC is a multi-protein complex. I told you these tritine family members act as a scaffold. So this STRIPAC is a tritine interacting protein phosphatase and kinases. So this is a multi-protein complexes where various phosphatases and kinases like PP2A and other kinases like MOB4, and they are present CCM3. So this is a single complex where stritins or the SG20 they act as a scaffold. So there are some study which show that the members of this STRIPAC complex, multi-protein complex like SL, MAP, sarcolemma membrane associated protein, they are present in the endoplasmic reticulum. And the role of this STRIPAC complex has been established in various diseases like neurodegeneration, cancer, and other disease. But in context of ER functioning or the ER stress, it has not been established. So this is our next objective, to find out the role of this STRIPAC complex and the member of this STRIPAC in the ER functioning. These are the references. Thank you.
Thank you, Dr. Buddhi Jain, for sharing a very valuable information regarding the role of SG2NA in endoplasmic reticulum stress. I hope the audience will benefit from the information they have got, and they will pursue similar kind of work in their respective areas. Thank you very much for your valuable information.
Thank you.